Coronavirus, Zika, Ebola, flu, even the boring old common cold. We are all familiar with the viruses that plague humanity, but while we know they make us sick, it may be surprising to discover that over millions of years, we have managed to harness and domesticate these crafty invaders. From the earliest stages of life to the smiles on our faces, viruses have had a huge influence on our human species. Viruses are little more than a string of genes, usually in the form of a molecule called RNA, packaged in a protein code, and they all work in the same basic way. Once a virus has infected a cell, it hijacks the cell's own molecular machinery to copy its genes and churn out vital proteins. New viruses are assembled from these freshly manufactured parts, which eventually burst out in search of new cells to attack. For most viruses, such as flu, the story ends there. But a handful of retroviruses, including HIV, are even sneakier. Smuggling their ways into our DNA, they insert themselves randomly into the genome of an organism, lying low until the time is right to start virus production again. But once a retrovirus has got into an organism's DNA, there is no guarantee that it will stay put. The genetic instructions can be read from the embedded virus, converted into DNA, and then passed into another location in the genome. Repeat this cycle again and again, and multiple copies of the viral DNA quickly build up. Over millions of years, these viral DNA sequences randomly mutate and change, losing their ability to break free from their host cells. Trapped inside the genome, some of these endogenous retroviruses can still jump around while others are stuck forever where they last landed. And if any of these events happen in the germ cells that makes egg and sperms, then they will be passed down the generations and eventually become a permanent part of an organism's genome. Around half of the human genome is made up of millions of DNA sequences that can be traced back to long dead viruses or similar jumping genes, known collectively as transposable elements or transposons. Some researchers even put this figure up to at around 80%, as ancient sequences are now degraded beyond the point of being recognizably virus-like, weathered within the genome like molecular fossils. For many years, the large chunks of repetitive virus-derived DNA littering the human genome were dismissed as junk. A proportion of this repetitive stuff undoubtedly is little more than junk in our genetic trunk. But as researchers look more closely at individual viral elements, a more sophisticated picture is emerging. And it turns out that as well as our genetic enemies, some of the viruses embedded in our genome have become our slaves. Syncytin evolution. Around 15 years ago, US researchers discovered a human gene that was only active in the placenta. They called it syncytin because it makes a molecule that fuses placental cells together, creating a special layer of tissue known as syncytin. Curiously, syncytin looks a lot like gene from a retrovirus. Another syncytin gene was later discovered, which is also involved in forming the placenta as well as preventing the mother's immune system from attacking the fetus in her womb. Again, the gene looks like it has come from a retrovirus. But while humans and other large primates have the same two syncytin genes, they aren't found in many other mammals with similar fused cell layer in the placenta. Mice also have two syncytin genes. They do the same job as human version, but they look like completely different viruses. And there's another separate virally derived syncytin gene in cats and dogs, both of which are descended from the same carnivorous ancestors. Clearly, all these mammalian species were infected by particular viruses millions of years ago. Over time, those viruses have been harnessed to play a key role in placental growth, making them a permanent fixture in our genome. Intriguingly, pigs and horses do not have a layer of fused cells in their placenta, and they also do not have any genes that look like virally derived syncytins, so maybe they never caught one of these fusing viruses. Jumping genes. While the case of syncytin reveals the wholesale adoption of a virus gene to do our bidding, there are many more examples of how ancient viral sequences can influence gene activity in today's humans. Back in the 1950s, Painstakingly detailed work by the long overlooked American geneticist Barbara McClintock revealed that jumping genes could affect the genome of maize plants. And just like jumping genes, McClintock identified in maize, the endogenous retroviruses that lurk in our human genome have been on the move over millions of years, jumping around at random and altering the activity of genes in their immediate vicinity. Our cells invest a lot of energy in attempting to stop these viral elements from going on the top. They are labeled and locked down with chemical tags known as epigenetic marks. But as the viral elements move, these molecular silences move with them. So the viral sequences effect can spread to neighboring genes wherever they land. Conversely, viruses also are full of DNA sequences that attract molecules which switch genes on. In a functional retrovirus, these switches activate the viral genes so it can become infectious again. But when a virus-like sequence gets spliced into another region in the genome, this ability to act as a genetic switch can end up going rogue. In 2016, scientists at the University of Utah found that endogenous retrovirus in the human genome, which originally came from a virus that infected our ancestors roughly 45 million to 60 million years ago, switches on a gene called AIM2 when it detects a molecule called interferon, which is the danger signal that warns the body that it is suffering a viral infection. AIM2 then forces the infected cells to self-destruct to prevent the infection from spreading any further. These ancient viruses have become double agents, helping our cells to tackle other viruses that are trying to attack us. Another example of a virus that may have shaped our species is found near a gene called PRODH. PROD is found in our brain cells, particularly in the hippocampus. In humans, the gene is activated by a control switch made from a long dead retrovirus. Chimpanzees also have a version of the PROD gene, but it's not nearly so active in their brains. One possible explanation is that an ancient virus hopped a copy of itself next to the PROD in one of our long dead ancestors millions of years ago, but that this did not happen in the ancestral primates that went on to evolve into today's chimps. Today, faults in PROD are thought to be involved in certain brain disorders, so it's highly likely to have at least some kind of influence on the wiring of a human brain. Similarly, variations in genetic switches are responsible for the differences between the cells that build our human faces as we grow in the womb and those of chimps. Although our genes are virtually identical to chimpanzees' genes, we certainly don't look the same, so the difference must lie in the control switches. Judging by their DNA sequence, many of the switches that are active in the cells that grow our faces seem to have originally come from viruses, which must have hopped into place sometime in our evolutionary journey towards becoming the flat-faced species we are today. The virus tamers. As well as searching for examples of long-dead viruses that have altered our biology, scientists are searching for the control mechanism that underpin their effects. The key culprits are special silencing molecules called crab zinc finger proteins, crab ZFPs, which grab hold of viral sequences in the genome and pin them in place. Professor Didier Trono and his team at the University of Lausanne in Switzerland have discovered more than 300 different crab ZFPs in the human genome, each of which seems to prefer a different virally derived DNA target. Once there, they help to recruit the molecular machinery that turns genes on or off. These crab ZFPs have been viewed as killers of these endogenous retroviruses, Trono explains, but they're actually exploiters of these elements that allow the organism to exploit the wealth of possibility that resides in these viral sequences. Trono and his team believe that crab ZFPs are the missing link between viral sequences that are actively harmful and those that have become tamed control switches. They have evidence that the protein have evolved alongside the viral elements in a kind of an arm race, initially suppressing them but eventually overpowering them. We think that they do is domesticate these elements, Chrono says, and by domestication, I mean not just making sure that the viruses stay put, but turning them into something beneficial for the host, which is a very refined way of regulating gene activity in all possible cells and situations. Supporting this idea is the finding that distinct group of crab ZFPs are active in different type of cells. They're also found in specific patterns in different species. If they were just spreading viruses, the argument goes the same array of proteins should be present in all cells. What's more, why would they be found bound to the many thousands of long dead viral elements that Toronto and his team have identified? There's no point suppressing a dead retrovirus, so they must be playing an important role in controlling gene activity. Although his idea is still a little controversial, Toronto sees the crab ZFPs as a force of viral slave drivers, harnessing these elements to do our bidding and turning them into genetic control switches. Over millions of years, this could have been a powerful motor for creating new species. For example, if a virus randomly goes on the hop in one ancestral creature and not another, and is then tamed over time by a crab ZFP, it will create new control switches that could have a big impact on an animal's appearance or behavior. What's more, these jumping elements become more active during times of environmental change. As times get tough, species need to find new ways to adapt or they will die out. Activating these mobile elements reshuffle the genome, throwing up novel genetic variations that provide richer fodder for natural selection to work on. Viruses, the good, the bad, and the beneficial. It's clear that the viruses trapped in our genome have brought us enormous benefits on an evolutionary timescale. But they aren't also helpful. Around 1 in 20 human babies is born with a new viral jump somewhere in its genome, which could deactivate an important gene and cause disease. There's an increasing evidence that jumping transposons contribute to the genetic chaos inside cancer cells. And intriguing research suggests that the brain cells are particularly good locations for reactivating jumping genes, possibly increasing the diversity of nerve cells and enhancing our brain power, but also potentially causing aging-related memory problems and conditions such as schizophrenia. So, are these viruses inside our DNA our friends or our enemies? Paolo Mita, a postdoctoral fellow researching transposons at NYU School of Medicine in New York, suggests that it's a bit of both. I call them our frenemies because when you look at their role in one human lifespan, most likely if they're mobilized, they're going to have negative effects. He explains. In the short term, they are our enemies. On the other hand, if you are looking across time, these elements are our powerful force of evolution, and they are still active in our species today. The evolution is just the way that organisms respond to change in the environment, and in this case, they are definitely our friends because they have shaped how our genome works now. And are viruses infecting us today, such as HIV, going to have an impact on our evolution in the future? Of course. The answer is why not, laughs Mita. But it will be many generations until we can look back and say this evolution has happened. But you can see the remnants of previous arms race in the genome between the endogenous retroviruses and the host cells. It is a continuous battle, and I don't think it has ever stopped. Says Mita.
Hi, I hope you're enjoying the videos. I'm not going to beg or persist or force you to subscribe or like